So let's talk through the rest of this chapter quickly. So, mitotic spindle, like I said, mitotic spindle is a structure made of microtubules that control chromosome movement during mitosis. So they are a very special kind of microtubules that we see during mitosis to control the chromosomes moving. So it's really important. And these are all organized and made in a place called the centrosome. Like I said, it's a microtubule organizing center. So it's where the microtubules are made, where they're directed, where they're destroyed, etc., etc. And the centrosome replicates during interphase just like the DNA does, and that way we get two centrosomes because we need to have one in each new daughter cell. And remember, they start off in one spot and they move to opposite ends of the cell, which are going to be where we pull the cell apart during anaphase. And an aster is a short array of microtubules that are kind of always growing out of the centrosome. So, the spindle makes up centrosomes, spindle fibers, and asters. During prometaphase, some spindle microtubules attach to the kinetochores of chromosomes. So remember, kinetochores are the protein structures that are at the centromeres of the chromosomes. And so the spindle fibers come out and they attach to the kinetochores, and then they pull them into the metaphase plate. So when they're lined up at the metaphase plate, one spindle fiber from each centrosome is attached to the kinetochore. So they're being pulled from opposite ends of the cell. So you can see right here, here's a kinetochore, and we have spindle fibers from this centrosome and from this centrosome. Okay, so we're not really sure how these microtubules get broken down, but basically, we don't know if they're being removed from this end or if they're being removed from this end. They don't know if they're being reeled in from the centrosome or if they're breaking off closer up to here. So this is a lot of research being done. We can talk more about that later. Okay, let's talk about the cleavage furrow. So when cytokinesis is happening, like I said, the actin ring forms around the cell and starts to contract and contract and contract and contract, forming two new daughter cells. And this is called the cleavage furrow. So, do you have a picture? So right here, when it starts to happen, that's called the cleavage furrow, until it eventually pinches off and forms two new daughter cells. Now, this isn't going to happen well in plant cells because they have a cell wall, and it's made to not do that. So what instead happens is, as mitosis is finishing up, the Golgi apparatus of each new daughter cell will start to send these vesicles made with cell wall components to the middle of the cell. And they'll just deposit them, and eventually we'll just send all these little pieces until we've built a new cell wall. So basically, here's the vesicles that are depositing the parts of the cell wall, and this is called the cell plate when it's forming. So this is the original parent cell, and we're building the cell wall in the middle, and they get dropped off by particles from the Golgi apparatus, which you can see here, until it actually forms a new cell wall. We saw all of these. Okay, so in prokaryotic cells, it happens a little bit differently because they don't have a nucleus, so they don't do mitosis. Remember, because mitosis is division of the cytoplasm. I'm sorry, division of the nucleus. So they instead use a process called binary fission. And so what happens is the chromosome replicates, because remember, we need a chromosome for each of the daughter cells, and it starts at a place called the origin of replication. So here is the parent cell, and here's the origin of replication. We're going to talk a lot more about origin of replications when we talk about DNA synthesis. But the origin of replication is right there, and it starts to divide, starts to replicate the DNA. And then the two chromosomes, what happens is the two chromosomes pull apart. So here we have chromosome replication beginning, and then it's continuing to replicate, but you can see that one origin of replication has moved to one side and the other has moved to the other side. So now we have two because we're replicating the whole piece of the DNA. And that's part of the DNA. And then eventually what happens is the origin of replications pull the chromosomes to opposite sides, then the cleavage furrow happens and we have two new daughter cells. So that's binary fission. You don't need to know this. Okay, one more important thing. We're going to go all the way back to the beginning of the section. we're going to talk about what happens during the during these 3 phases. So these are all interphase, right? So this is when the cell is just doing its job. Basically, during G1 phase, the cell is growing. It's doing whatever job it's supposed to be doing, and prepares for DNA synthesis. So G1 is growth and preparation for DNA synthesis. S phase is synthesis of the DNA. G2 phase is growth and preparation for mitosis. That's basically all you need to know.